Hello everyone and welcome to video 9. So we should know from the last video tutorial that enzymes speed up chemical reactions and the substrate has to be a complementary shape to the active site of the enzyme for it to react. And we should also know that there's two types of reactions that the enzymes are involved in, one of which is degradation, and you should know the 4 different types of degradation reactions that you get, what the substrates are, what the enzyme are, and what the products are, and same with your one synthesis reaction. Also, we should know that enzymes are made of proteins. So what we're going to move on to is how temperature and pH affects enzyme activity. So you can go print off the consumables from activities and consumables in 1 cell biology and then in 4 proteins, and then in 4.3 temperature and pH effect on enzymes. So for your starter activity you have this question. So it says the diagram below shows how the enzyme lactase is used in the production of lactose-free milk. And then you've got your milk containing lactose, and then you've got jelly beads with lactase enzyme attached. And then you've got the lactase splits the lactose into small sugar molecules. And then you've got your lactose-free milk in your beaker. So it says underline one option in each of the brackets to make the following sentence correct. So this process is an example of either a degradation or a synthesis reaction. And in this reaction, lactose is the product or the substrate of lactase. So you guys want to try to answer the question and before I reveal the answers. So you can pause the video and try and answer it. So for to answer this, we need to know what a degradation and synthesis reaction is. So we can read in this passage here that the lactase splits the lactose into smaller sugar molecules. So if it's going from a big molecule, which is attached, into a smaller molecule, then it's going to be a degradation reaction, a breakdown reaction. And in this reaction, lactose is it's lactose that's formed at the end, so it's going to have to be the product, because remember the substrate is at the start. So if you managed to get that all correct, well done. If you didn't manage to get that all correct, then you need to go back and watch video 8 again. Your learning intention today is to know how temperature and pH affect enzyme activity and to be able to describe it, to know what optimum means, and to describe what is meant by when an enzyme becomes denatured. So to be successful at this, you'll be able to explain what happens in a temperature graph and a pH graph. You'll be able to identify an enzyme's optimum pH and temperature. You'll be able to explain what happens when an enzyme becomes denatured, and you'll be able to answer questions about enzymes. So there's 2 factors which affect enzyme activity, which we've already mentioned. So an enzyme is most active with a suitable temperature and pH. So if your temperature is too high or too low, your enzyme will not function properly. And for pH, if your pH is extremely acid or extremely alkali, then your enzyme will not function properly. So you'd want to write a note on this, so you'd put optimum conditions for enzymes as your title. So each enzyme is most active in its optimum condition. So optimum is where your enzyme is most active, and you need to know this word optimum and what it means. So enzymes and other proteins can be affected by temperature and pH. So your optimum condition is where your enzyme is most active, and that is what you need to pick up from here. So enzymes have an optimum, and optimum means that your enzyme will be most active. So your enzymes are proteins and they can be affected by temperature and pH. So you can measure enzyme activity in a variety of different ways, and it also depends on the enzyme that you are looking at, because we obviously know that there's lots of different degradation reactions and there's one synthesis reaction that we need to know. So for this, we are looking at hydrogen peroxide. So this can be a little test. So hydrogen peroxide would be your substrate, which would be, so Harry Potter's, and then we would want our enzyme, which would be our catalase, so cat, and then we would have our product, which would be our water, well, our product, which would be our water and our oxygen. So Harry Potter's cat went out. So there are two experiments which are shown here. This one here and the second one, which is over here, which both measure the enzyme activity. So they're both measuring the catalase activity in different types of tissue, but they were both doing it in different ways. So for the first one, it's measuring the volume of oxygen gas collected in 3 minutes. So they've got a measuring cylinder with its delivery tube. So you've got your hydrogen peroxide and your cube of potato, which contains catalase, or your cube of whatever tissue it would be exchanged for. And then they're measuring the volume of oxygen gas collected in 3 minutes. And they're doing it 4 times and then taking an average. Now this measures the catalase activity because we know that when our catalase is present, it speeds up your hydrogen peroxide breaking down to water and oxygen. So the bit that we are measuring is how much oxygen is produced. So the more oxygen produced, the better the catalase activity. So the higher the value, the better it will be. The second diagram is slightly different. So what is happening in this one is you've got filter paper and it is soaked with either apple, banana, carrot, liver, onion or potato. And then it's put into your hydrogen peroxide and then it is timed for how long the paper disc takes to go down into the hydrogen peroxide and return back to the surface. Again, this is measuring oxygen but via a slightly different method. So basically, if there's lots of oxygen bubbles, then the paper disc will go faster to the surface. So this time, the lower the number, the more oxygen produced, so the more enzyme activity, so the better catalase activity there will be. So that makes a slight difference because for experiment 1, the higher number, the better the catalase activity. But for experiment 2, the lower the number, the better the catalase activity because the more oxygen produced in each of these experiments. Now, for experiment 1, this can be also carried out, that experiment, but slightly differently. If you get hydrogen peroxide and you put your tissue in it, then you can measure your height of your oxygen bubbles by trapping them in fairy liquid. And the second experiment can be done but using something called immobilized beads. So you can trap your tissue in immobilized beads, but it's exactly the same idea. So you'd put your immobilized bead in and then you'd time how long it took to resurface. So both experiments are measuring the oxygen level and how much oxygen has been produced, and both are measuring the enzyme activity, so the catalase activity, but they're completely different methods and you get completely different results, so be careful when you're looking at enzyme results. So if we try to calculate the average of the potato, so then we can produce our conclusion. So to calculate an average, we know that we find the total and then we divide it by the number of groups or sets there are. So we would want to add 5.5, add 5, add 5.5, add 6, and then there is 1, 2, 3, 4 groups. So we press equals and we divide it by 4, which would give you the answer of 5.5. So for the first experiment, the conclusion would be that the liver had the highest catalase activity, and that's because the volume of oxygen collected in 3 minutes was highest for the liver. It was about 30 times higher in some cases than the other tissues, or you could say that animal tissue seems to have a higher catalase activity activity than plant tissue, so either conclusion would be acceptable. For the second one, you would want to say again that liver has the highest catalase activity because it only takes 8 seconds for the disc to return to the surface, which is much higher, much faster than your other tissues, so your plant tissues. Or you could say that your animal tissue was the fastest at returning to the surface compared to your plant tissues, and you could work out how much times faster it was than your slowest tissue. So that would be 108 divided by 8. So that would give you 13.5. So the liver was approximately 14 times faster at rising to the surface than the apple. So that suggests that there was more oxygen produced by the liver, so suggesting that it had more catalase activity. So animal tissues have higher catalase activity than plant tissues. Now, you would usually do these experiments, or at least one of these experiments, in class. You probably will still do one of these experiments when you get back. So another way you can measure enzyme activity is by a gel. So for example, if we make up a starch gel, we could find out what enzyme is most effective on the starch gel, or we could do various pHs, etc. So if we, for example, take the different enzymes, so if we put amylase in a well here, and then we put catalase in one of the other wells, lipase in another, and protease in the last one. What we would expect to see if we left it for like overnight is there should be a clear zone that starts to form around amylase, but none of the others should form this clear zone. And that's because if you remember, and this should be revision, that starch is the substrate of the enzyme amylase, and amylase speeds up the reaction of starch to glucose. So measuring this clear zone gives you an indication of enzyme activity. So another experiment that you would perform in class is a temperature investigation. So you would look at a tissue in hydrogen peroxide at various different temperatures. So if you're doing an experiment, it's important that you only change one variable. So our aim is to investigate if temperature affects catalase activity by measuring the height of oxygen bubbles. So what you would do first of all is you'd pick a specific tissue, so it might be potato, it might be liver, and so on. So we picked potato this time, and you need to make sure you've cut them to equal sizes because you need to control all of your variables except from one. So your potato cylinders need to be equal sizes and the same type of shape. Then you've got to blot them dry to make sure there's no excess water before you place them in the hydrogen peroxide. You would need to leave them at the temperature for the same time, and you would put them in different water baths. So you'd have one water bath that was set to 0 degrees Celsius, you'd have another water bath set to around 20 degrees Celsius, another one set to around 40 degrees Celsius, and another one set to around 60 degrees Celsius. So then you would leave them for like 5 minutes. So after 5 minutes, the following was observed. So you've got your 0 degrees Celsius is quite low with oxygen bubbles, 20 is higher, and 40 is your highest, and 60 there's no bubbles. So you could try to make a conclusion for this, but you don't really know how or what's happening. All we know is 40 degrees Celsius is giving us the highest catalase activity, and we need to know why that is. So we're going to look into that into more detail so we can come up with a proper conclusion. So to make a conclusion, we need to know how temperature affects enzymes. So as you vary the temperature that a reaction takes place at, you vary the energy involved. So the higher the temperature, the more kinetic energy, so the more your particles move and bounce off each other. And you learn in chemistry that basically the higher the temperature, the faster the reaction rate. So low temperatures mean that the molecules have little energy, so there's little kinetic energy, and high temperatures means that they have more energy, so there's going to be more chance of them being able to collide. So if you imagine if you had like 4 balls and it was really, really cold, they'd be moving really, really slowly, so the likelihood of them colliding with each other if balls are moving slowly is very, very not likely. Whereas if you've got a hot temperature and your balls are moving really, really fast, there's more chances of them hitting into each other. So we know that for reactions to happen, our particles must collide together. If they don't collide together, then the reaction can't take place. So in the National 5 Biology, you will probably be asked to describe a temperature graph. Okay, so you'll have your enzyme activity and you'll have your temperature degrees in Celsius. And you'll be asked to say what is happening at each stage. This could be a 4-mark question, or you could just get asked what the optimum is, or you could just get asked what's happening at each section. So you have section A, section B, section C, section D, and section E, which I'm going to talk you through. So for section A, we notice that A doesn't actually go to the origin. It starts slightly further up, and that's because the enzymes are not damaged, so they're still functioning at low temperatures. Okay, so at low temperatures, enzyme activity is slow, and that's because the molecules have less kinetic energy, so there's going to be less collisions. So in the case of B, your temperature started to increase. So as your temperature increases, your enzyme activity increases, and this is because molecules have more kinetic energy, so they move faster and will collide more. So in B, you've got a steeper slope, which means that you've got faster enzyme activity as you've increased the temperature. So this might be about 10 to about 20 degrees Celsius, and your enzyme activity is increasing, and that's because the molecules can bounce off each other and collide. So for the next part, we hit a kind of peak. So for this bit, we're referring to this bit here, which is C. So at C, we hit a maximum, and the maximum enzyme activity we referred to, if you remember at the start, is its optimum. So around about 35 degrees Celsius to 40 degrees Celsius. So usually 37 degrees Celsius is the temperature at which this occurs, and it's known as the optimum temperature because the enzyme activity is at its maximum. So above the optimum, we start to see a decline, so quite a big decrease. And that's because as the temperature becomes greater than the optimum, the enzyme structures start to change. So the shape of the active site changes and loses the ability to fit with its substrate. And when this happens, enzyme becomes denatured. So it's when the active site changes shape and can no longer bind to its substrate. This is called denatured. So we know that enzymes are made of proteins, and proteins are held together by bonds. So if you heat the temperature above its optimum, then the enzyme's bonds and structure will break and change, which causes the active site to change shape, which will mean that the substrate no longer fits, and that's called denatured. So the last part, E, so as the temperature increases, more and more enzyme molecules are affected and become denatured, and the reaction stops. So you need to be able to say what's happening at A, B, C, D, and E. So you need to be able to describe this whole curve. So you need to be able to say what's happening at A, B, C, D, and E. So you might want to pause the video. You might want to write out what's happening at A, B, C, D, and E until you know it off by heart. So obviously at low temperatures, the enzymes are not damaged, but they're quite inactive. So basically they move very, very slowly because there's not a lot of energy, so there's less collisions. And then as the temperature increases, there starts to be more and more energy and more collisions, so that's why there's an increase at B. And then we get to the enzyme's optimum at C, so the part where the enzyme works the best, the enzyme's most active. Okay, so optimum, enzyme most active. And then at above the optimum temperatures, the enzyme's activity starts to decrease quite steeply, and that's because your enzymes start to denature. So the active site of the enzyme changes shape and can no longer fit with your substrate, and more enzymes become denatured, and eventually your enzyme activity stops altogether. So if you don't know that, remember you need to go over and over it until you do know it and until you can move on. So there's one exception to this curve, which obviously shows as the temperature increases, enzyme activity increases. If you measured your time in minutes and instead had time in minutes on the axes and temperature for your filter paper to rise or your immobilized bead to rise when you've got your tissue trapped in your immobilized bead, or you've got your tissue wiped onto your filter paper, you'd get a slightly different curve. So what would happen is you'd get it upside down. So basically you would have your fastest time at the bottom, which would be your optimum, so that would be your C, and this would be your like a slower time. This would start to become faster, this would start to slow down again, and then this would be the slowest time of all of them. Okay, so just being careful for what you're measuring. Enzyme activity will always look like this curve, but if you've got your time in minutes, it might look slightly different. So just a warning about that. So we've looked at how temperature affects enzymes. So this is just a summary. So at very low temperatures, your enzyme is inactive but undamaged. At low temperatures, enzymes and substrate molecules move slowly, so there's not very many collisions. As temperatures increase, your molecules move at a faster rate and your reaction rate increases because there's more collisions. And most suitable temperature would be your optimum. So the one where your enzyme is most active is your optimum temperature, and the reaction rate is at its peak. So it's usually 37 degrees Celsius, but it's usually between 35 and 40 degrees Celsius. So above your optimum temperature, so usually above 40, your active site is altered and your enzymes no longer fit with their substrate, and your enzyme becomes denatured. At very high temperatures like 55 to 60 degrees Celsius, your enzyme activity completely stops because all of your enzymes are then denatured. So optimum conditions for enzymes. So enzymes usually work best at 37 degrees Celsius in humans, and that's because that's body temperature. So this is a curve for if you have enzymes in the body. Now, for if you wanted to say about your optimum temperature, you could just say that usually the optimum temperature for an enzyme is 37 degrees Celsius, and you would get away with that. You don't want to say that it's always 37 degrees Celsius because that's not necessarily always correct. So usually your optimum temperature would be 37 degrees Celsius. So as you increase the temperature, there's heat energy causes more collisions between your enzyme and your substrate, which is going to increase your rate of reaction. And then at 37 degrees Celsius, that gives you, if you go down here, you can always find your optimum by dragging the line down with a ruler, and then here would give you 37. So that would be how you would measure your optimum from a graph. And then above the optimum temperature of 37 degrees Celsius, your enzymes denature at high temperatures, so the reaction rate falls rapidly, which is what we've seen and said in the last part of this video tutorial. So what does it mean by denatured? So enzymes can be denatured at high temperatures, resulting in a change in the shape of their active site, which will affect the rate of reaction. so as they can no longer bind to their substrate. So you need to take a note of this. So put temperature and denaturation. So at temperatures above the optimum temperature, enzyme molecules start to denature. So this is just an illustration of what would happen if your enzyme was put into boiling water or any high temperature. That's usually about 60 degrees Celsius. So if the enzyme is placed in boiling water, the active site of the enzyme changes shape. So the substrate molecule that used to be able to fit into the enzyme's active site can no longer fit into the enzyme's active site. So basically the enzyme can no longer work. So the change is permanent and we say that the enzyme has now been denatured. So going back to the experiment of the temperature investigation. So obviously the aim was to investigate if temperature affected the catalase activity by measuring the height of the oxygen bubbles, and we had these results. So you're going to try and make your own conclusion from what we've just learned about temperature, and you're going to pause the video and then I'm going to give you the answer. So you would have had to write this for your conclusion to get your mark. So as the temperature increased, the catalase activity increased until it reached the optimum temperature at 40 degrees Celsius. Above 40 degrees Celsius, the catalase activity decreased as the enzymes became denatured. So you need to say, first of all, the temperature increasing is causing the catalase activity to increase. You could have mentioned that's because it increases collisions. And you needed to say that it was until it reached the optimum temperature, which in this case was 40 degrees Celsius. But for the next part is really important. You need to mention that above 40 degrees Celsius, the catalase activity decreases as the enzymes become denatured. So you needed all three different parts. So the trend that it increased and then it reached its optimum and then it decreased to get your marks. So here is an example of an exam question. So the optimum temperature for the activity of catalase is 37 degrees Celsius. Predict what would happen to the activity of catalase if the temperature was lowered to 34 degrees Celsius. Well, we know that if you reduce the temperature, there's going to be less collisions, so there's going to be a reduce in catalase activity. So you would want to write that catalase activity would decrease. If this question had said, predict what would happen to the activity of catalase if the temperature was increased above 37 degrees Celsius, again you would want to put catalase activity would decrease, but this time instead of because there was less collisions, it would be because the enzymes started to denature. Now, if you had an experiment that was carried out at 20 degrees Celsius for an enzyme, and you didn't have what the optimum temperature was, and it says predict what would happen to the activity of the enzyme if the temperature was increased to 37 degrees. You would then put that the enzyme activity would increase because it would be nearer the enzyme's optimum, which is the enzyme's most active conditions. So here is another type of exam question that you're likely to face. So it says, explain why high temperatures above 50 degrees Celsius would prevent the photosynthesis reaction from taking place. Now this might be photosynthesis, it might be respiration, or it might be something else that's controlled by enzymes. So there's two things that this question is looking for, so it would be a 2-mark question. The first thing it's looking for you to say is that, and recognise, is that this process, so photosynthesis, is controlled by enzymes. And the second thing it's looking for you to say that is that above 50 degrees Celsius, the enzymes would be denatured because the active site of the enzyme would change shape and would no longer bind to its substrate. So enzymes are also affected by pH as well as temperature. So you need to make a note on pH. So pH is measured on a pH scale. So you should have tested things for pH by using universal indicator, and we know that things below 7 are acid, things that are 7 are neutral, and things above 7 are alkali. So enzymes are active over a narrow range of pH values, and that would be where your enzyme is active. There'll be an pH that your enzyme will be most active and a pH where it'll be least active, and the bit that it works over will be called its working pH range. So within the working pH range of the enzyme, there will be a pH at which the enzyme is most active, and this will be called its optimum pH. So remembering the word optimum again, so optimum meaning most active. So just like temperature, pH also has a graph of what happens with enzyme activity. However, different enzymes have different pHs that they work within and different optimum pHs, whereas usually temperature is more across the board for enzymes, but not always. So if you got asked these questions, you should be able to answer them, and you want to take a note of this. So the enzyme is active over the range, and then it would be from this value, so it's its working range, from 6 to 8. So above this or below this, the enzyme is not active. And to find optimum pH, it's just like trying to find the optimum temperature. You'd get a ruler and then you would go down. Obviously, I don't have a ruler, so it's a little tricky to make these lines straight, but you'd get a ruler and you would end up at the 7. So the optimum pH of this enzyme, so where it is most active, so the top of the peak would be at 7. So we would have 6, 8 and 7. So this is specific to a specific enzyme. Different enzymes would have different working ranges and different optimums. So different enzymes work within different ranges. So if you look at pepsin, for example, its working range is about 0.8 and to about 5. And then if you want to look at the optimum, we always need to take it from the top of the peak and then go down with a ruler. So that gives us about 2.8. Then our optimum for amylase, which is more neutral than the acidic conditions that pepsin work in, would be, the working range would be from 5 to 9. And then if we take the top of the peak for amylase, we get 7. Then our optimum for catalase, which is more alkaline, is our pH of 9. And our working range would be between 6 and 12. So as you notice, all of these different enzymes have different working ranges and different optimum pHs. Most enzymes work in a narrow range around pH 7 neutral, for example, amylase. Some enzymes work best in acidic conditions, so for example, pepsin in the human stomach. We know the human stomach contains hydrochloric acid, so its optimum and working range of pHs is around 2 to 3. And then pH values below or above the optimum pH can cause the active site of enzymes to change shape because the extreme acid or alkaline conditions causes the bonds to break in the protein, which causes that active site to change shape, and then the active site can no longer fit with the substrate because it's no longer complementary. So then the enzyme is said to be denatured. So each enzyme has a pH it will be most active at, so that would be its optimum pH. Pepsin works in the stomach, which is acid. Amylase works in the mouth, which is neutral. And catalase works best in alkaline conditions. So optimum conditions for enzymes pH. So as we've said, pH changes can also lead to denaturation of enzymes. So you've got your substrate, and usually your substrate will fit with your active site of your enzyme because they'll be complementary to each other. But if you put it in a pH of a solution that's very acidic or alkaline, so extremely acidic or alkaline, then the bonds of the protein break and the active site of the enzyme changes shape. So the active site of the enzyme is no longer complementary to the substrate, so the substrate will not fit into the active site of the enzyme. So there's a little video to recap. You can't click on the video tutorial, but if you go to the PowerPoint presentation, you'll be able to watch it. For to review this video tutorial and also 8 and 7, you can now complete the cell biology exam papers, which is found in Teacher's Guide and cell biology exam practice papers. So you would do page 14 to 18. You can complete the DNA, protein and enzyme past papers, which is found in the proteins, and then it should be past papers. And then you can complete the National 5 textbook testing your knowledge questions on page 38 and 42. And then you can complete the How to Pass National 5 Biology questions on page 22. Both of these you can get from the textbooks. And then you should then be able, after you've done all that, to complete the homework activity. And once you complete the homework activity on enzymes, you then send it to your teacher for marking, so we can make sure that you're understanding the work. So well done for getting this far. We're going away to look at some exam questions in the next few slides before we finish. So to be successful, you should now be able to know that there's two things which affect enzyme activity, and these two things are your temperature and your pH. You should be able to describe what happens to enzyme activity when you increase and decrease the temperature. You should be able to know what the word optimum means and how you'd find the optimum temperature. You should be able to know how pH affects the enzyme activity. You should be able to know what working range of pH is and how you would get the working range of the pH, and you should be able to know that what the optimum pH is. You should know what the word denatured is and how it applies to enzymes after they've been in either a temperature above their optimum or an extreme pH of either highly acidic pH or an highly alkaline pH. So if you are able to do all of that, great. We're going away to practice some SQA questions. If you're not able to do that, then you need to go back and watch this video again. So for these questions, I'm going to go through the questions first and then I'll go through the answers. So for question 1, enzymes act as catalysts because they A, are composed of proteins, B, act on any substrates, C, are unaffected by temperature, or D, speed up chemical reactions. So is it A, B, C, or D for question 1? So for question 2, the enzyme catalase breaks down peroxide into water and oxygen. The experiment was repeated at 5 different temperatures and the results are shown in the table below. Identify the temperature at which the catalase was the most active and explain why no oxygen was produced when the investigation was repeated using a different enzyme. So A, you want to identify the temperature at which catalase was the most active, and B, explain why no oxygen was produced when this investigation was repeated using a different enzyme. 3, the active site of the enzyme is complementary to A, one type of substrate molecule, B, all types of substrate molecule, C, one type of product molecule, or D, all types of product molecules. 4, all enzyme molecules are composed of A, carbohydrates, B, glycerol, C, proteins, or D, fatty acids. So for 5, give one condition which could cause an enzyme to become denatured. So give one condition which could cause an enzyme to become denatured. So let's have a look over the answers. So for enzymes act as catalysts because it would be speed up chemical reactions. For 2A, identify the temperature at which catalase was the most active. So we need to look at where the increase in oxygen percentage is the highest. So it should then be 35 degrees Celsius. Remember to always include your units of degrees Celsius. And explain why no oxygen was produced when the investigation was repeated using a different enzyme. Well, we know that enzymes are specific to only one substrate because the active site will only fit in one substrate. So the active site of an enzyme is complementary to one type of substrate molecule, and all enzyme molecules are composed of proteins. And give one condition which could cause an enzyme to become denatured, so that would be high temperatures or extreme pH. So that's the end of our video tutorial on enzymes, woohoo, and the end of key area 4. So next we'll be moving on to key area 5, which is genetic engineering. Well done for getting this far, guys.